Hey everybody. So this is chapter 3, the first recording of chapter 3. You will have a test on chapters 2 and 3, probably Wednesday. So you need to be studying. Don't forget about chapter 2 while you're focusing on chapter 3. It'll just be one test with both chapters in it. So this is the chemistry of organic molecules. This is the last chemistry chapter before we actually get into biology. So 3.1 is organic molecules, just like an overview. Here are 3 definitions that you need to know. Remember, we're writing it in shorthand if we can. So organic chemistry is the chemistry of living organisms. Okay, if it's living, it's organic. And if it's inorganic, that means it's non-living. Okay, that means like non-living stuff or non-living matter. And then organic, the term organic actually means molecules and compounds that contain both carbon and hydrogen atoms. So a lot of the times we think of organic as being like the food that's not supposed to get sprayed with, pesticides and all that stuff in the grocery store. But the term organic in science actually means molecules or compounds that contain carbon and hydrogen. That makes them organic. All right, so the carbon atom is really important in chemistry and in organic chemistry. So if you looked at on a periodic table, you would know that the atomic number of carbon is 6. That is something that you should know. I should be able to ask you that on a test, and you know it. I also should be able to ask you that it always forms covalent bonds. Don't pay attention to the almost. We're not going to play with almost in biology class. Remember, covalent is sharing is caring. Caring starts with a C. So that's when we share the bag of chips or share the electrons. So a carbon-carbon bond is so stable that it allows the formation of long chains or even rings. So you'll see down there, like, let me see, that's 1, 2, 3, 4, 5, 6, 7, 8. That would be C8, H18. I'm not sure what that compound is called, but that would be the formula. And each little line represents a covalent bond of them sharing a pair of electrons. Okay. It can also form ring structures when it's placed in water. Double bonds between carbons aren't as flexible. They're not going to be as flimsy and move around in rings, so they restrict movement. But carbon can also form triple bonds within itself. Carbon's a big deal in organic chemistry. So those of you that say you want to be a doctor or go in the medical field, a pharmacist, veterinarian, anything along those lines, you're going to have to take chemistry, organic chemistry, inorganic chemistry, all that good stuff, and it deals with all of this carbon stuff, I guess I should say, all this chemistry stuff. So if you like that, that's what their background is in. So they call it a carbon skeleton, and then you add functional groups to it to make it different. So this is a skeleton or a backbone, and then the functional group definition is a specific combination of atoms that always have the same chemical properties regardless of the skeleton it's attached to. So you might have an alcohol that's attached to it, or a carboxyl group, or things like that. Here is some of the functional groups. You don't have to know these, but I just want you to be familiar with what a functional group is. The R on the screen represents the carbon skeleton. So a hydroxyl group is an OH, which makes it polar, because remember the oxygen is going to kind of pull those electrons a little bit. You're going to have a methyl group, which is CH3. It's going to be nonpolar. You got a carbonyl. I mean, this is stuff that you like get into deep. My minor's in chemistry, and I don't like chemistry, but some people do. If you do decide to take a chemistry, you'll go with Miss Brown. She's a really great teacher, and you guys will dive into this kind of stuff. All right, so this is a word that you're going to get mixed up with one from last chapter, which is isotope. Remember, isotope is when it's just different forms. They're going to weigh different, right? Like carbon-13, carbon-12, carbon-14. The weight of an isotope is different. An isomer, isomers are identical in formula, but different arrangements of atoms. So look at this picture right here. Butane has 4 carbons, and isobutane also has 4 carbons, and they both have the same amount of hydrogens. I'm not going to count them. But so their formula would be I guess I'll count them. It'll be C4H10 for both of them, but their arrangements are different. So that's what an isomer is. I don't know how, you can come up with something to figure out the difference between isotope and isomer for your test, like a little saying or something to help you remember it, but please don't get those confused. All right, so biomolecules of cells. Polymers. The prefix poly means what? Like a lot or many, right? So polymers are a large number of the same subunit linked together, like cars, proteins, nucleic acids, they all do this. Lipids are made of different subunits, so they are not polymers. Polymers, it has to be the same. Okay, so y'all write down what a polymer is. This chapter is not necessarily like last chapter. Last chapter, you have to know a concept of what types. You have to be able to figure out what kind of bonds, 2 atoms are going to make together, and you have to be able to draw Bohr models. This is basically just information. All right, synthesis and degradation. So when you build something up, dehydration reactions happen. So what happens is you're going to kind of suck out the water, I guess you would say. So you're going to dehydrate something and make it come together. If you dehydrate something, you're going to suck the water out, right? So to break down a molecule, those are called hydrolysis reactions. And think about hydro, you add the water back in and it kind of like dissolves that element or compound or molecule, okay? So if you want to put something together, you suck the water out, you dehydrate it. Those are degradation reactions. And then if you want to break down something, think about when you want to break down, I'm trying to think of something in the cooking world here. Like when you have something that's super thick, maybe you're making biscuits or maybe you're making brownies or something, and your batter is super thick. So what do you do to break it down and to kind of dilute it? You add water, right? That would be kind of like a hydrolysis reaction or a synthesis reaction. Enzymes can sometimes act as a catalyst for these to occur, to kind of like boost it up. Kind of like an energy drink, like get it started. Okay. So that was the overview. Let me see how many more we're going to today. We're going to do carbs and lipids, okay? And then the next Tuesday we'll do proteins and nucleic acids. And then I lied because we're off on Monday. So your test will be Thursday. So we'll have a review day. All right, so carbs. Please don't think of carbs as bread. Okay? I know it is bread, but we're looking at the molecular structure of it. So carbohydrates are the immediate energy source in living organisms. The ratio for carbohydrates is 1 carbon to every 2 hydrogens to 1 oxygen. So carbohydrates are made of carbon, hydrogen, and oxygen. Okay, if I had a board, I would write CHO. And if I had 12 carbons, I would have 24 hydrogens and 12 oxygens. Okay. The chain length will vary. What is its monomer subunit? Mono means one. What is it made of? It's made of monosaccharides. And then it has long chains of those called polysaccharides. Okay, so when you see the word saccharides, that means it's a carb. I will say, what is the monomer of a carbohydrate? I will say, what is the ratio to carbon to hydrogen to oxygen for a carbohydrate? That I 100% will put that on your test. So if you miss those, that's 110% on you. All right, so what is a monosaccharide? Remember, these are the monomer that makes up carbohydrates. These are simple sugars, kind of like sugars that you find in candy bars or a Mountain Dew. The carbon backbone is of 3 to 7 carbons. You'll have things like pentose, hexose, and glucose, ribose, and deoxyribose. Those are all monosaccharides, and then you just put a bunch of them together to make a polysaccharide. So what is a disaccharide? The prefix di means what? 2. Okay, so it's 2 monosaccharides put together. An example of this is sucrose. This is what we use sucrose to sweeten our food. And another example of this is found in milk, is lactose. So you'll hear a lot of people are lactose intolerant. That means their body does not have the enzyme to break down the lactose, so they can't have any milk products. All right, so we went from monosaccharides, which is 1, to disaccharides, which means 2, linked together. Now we have polysaccharides. So these are a bunch of sugars linked together. Okay? These are called complex carbs. This is like your pastas and stuff like that. This is actually a bread, or the breads you're thinking about. So in plants, these polysaccharides are called starches, and in animals, they're stored as glycogen. I'll ask you that too. All right, so another note is, you can just write it out to the side of your notes if you want to. I forgot to tell you, these two, starch and glycogen, are how you store. These are storage molecules. And then the next set of examples are structural molecules. So your body also uses polysaccharides or carbohydrates for structure in the cells as well. So for plants, that structural molecule is called cellulose. This is like wood and cotton. So that's why, how cool would it be if we had the enzyme cellulase in our stomachs? We could eat cotton. So look at your shirt right now. Most of your shirts are probably made of cotton. We could eat that if we had an enzyme in our stomachs called cellulase that would break it down. And we could eat wood too. And we would think nothing about it because we eat fruits and vegetables that grow just like it, and we don't think anything about it. How weird would that be to eat like bark? So the structural molecule for animals and fungi is called peptidoglycan. And chitin, that's how I'm going to pronounce it, is found in the exoskeleton of insects and crabs and things like that. So that's why we can't eat, we don't have that enzyme either. We can't eat, it's not good for our bellies, I guess I should say, to eat like the shell of a lobster or the shell of a crab. Although some people eat the shell of insects sometimes. All right, lipids. I know I'm going pretty fast. You can pause me if you need to, but when I go over a certain time limit, it won't allow me to upload it. So y'all can pause me if needed. So the last section we're going to go over today is lipids, and lipids are your oils and fats. So these are fats or triglycerides. When you get older, you'll have to do like health screenings and stuff, and they might say, oh, your triglycerides are high. You're going to have to, eat some Cheerios and get it down. They're talking about your lipid content. So we use lipids for insulation. So think about the fat cells on your body. And we use it for long-term energy storage. Your body stores up the excess food that we eat in fat cells, and so we can use it later and it can keep us warm. In plants, they're also referred to as oils. So what is a triglyceride made of? It has a fatty acid. So each has a long hydrocarbon chain with an even number of carbons. I will ask you the two parts of a triglyceride, which is a fatty acid and a glycerol, okay? Then I will ask you what's the difference in a saturated fatty acid and an unsaturated fatty acid. So there's two test questions that will come off this slide. A saturated fatty acid has no double bonds within its carbons, okay? It's saturated completely with hydrogens. If I had a board, I would show you. An unsaturated fatty acid has a double bond between two carbons. The unsaturated fatty acids melt faster than the saturated ones. I will ask you the difference between those two, okay? So two test questions from this. This is a college course, you do not get a study guide from me. You make your own notes. You have a book, you have me recording this, you have the PowerPoint. I'm not making you a sheet that says this is exactly what's on the test. But this is your first test, so I'm giving you hints throughout. Two questions. What are the two parts of a triglyceride, and what's the difference between saturated and unsaturated fatty acids? Okay. Pay attention to this. If I had a board, I would draw it because I feel like you guys pay attention to more of that kind of stuff. But because I don't have a board, look at the top picture. The top triglyceride, you can see the glycerol is over there in the red on the left. Then the long chain of carbons, the top one is a saturated one. You can see all the single bonds between the carbons. And the bottom one on the top picture is an unsaturated fatty acid or unsaturated triglyceride because it has a double bond within two carbons. Okay? You don't have to pay attention to the bottom picture is just showing you how the glycerol attaches to the three fatty acids. There are three fatty acids, hence the term triglycerides. I think we're almost done. 2 more slides. All right, phospholipids are really important in biology because this is what makes up the barrier of your cell, like the membrane of your cell and your organelles. Please remember phospholipids. Triglycerides, except in the place of the third fatty acid, there is a phosphate. So it's kind of like a triglyceride, but you have a phosphate group attached. And I don't want you to dismiss this by any means, but I will go over this further when I try to come there next week to review you. So it has a little polar head and not 2 nonpolar tails. Remember, it has 2 tails instead of 3 because it only has 2 fatty acids. So remember, fatty acids are like lipids or oils, so they hate water. So it's going to have a hydrophilic head and a hydrophobic tail. Hydrophilic means I like water. Hydrophobic means I hate water. So what they do is they kind of line up in this double membrane. You can see down there on the right side. And that's what makes your membrane, okay, for your cells. Steroids, I will ask you one question, I think, from this. slide, I will ask you a question, by the way, from phospholipids too. Steroids are lipid structures with a skeleton of 4 fused carbon rings. 4 of them. Cholesterol provides stability to cell membranes and is the precursor for to other steroids. So it's kind of like cholesterol is kind of stuck in the cell membrane to give it some support. Okay. These are things like testosterone, estrogen, all kind of cortisol, all kind of steroids. But look at the picture. Everybody look. Every single steroid has 4 carbon rings. Okay? And that is all the notes for today. Please check your homework. You will have homework. There's a 3-day week or 4-day weekend. You will have a sheet of homework. If you don't do it, I don't give you credit if you turn it in a week later, okay? Quarantine or not, you've got to be checking this.